Hello everyone. Let's start. So today we would be discussing cloning, PCR primers, a recap of that, and the DNA sequences as well. I hope it's visible to all. So first of cloning DNA in details. So basically, what do we want to clone here? We do have a source DNA of which we do want a particular target portion of that DNA, obviously not the whole DNA. Then we use what? A cloning vector, which is basically you can consider it as a vehicle or something. And so we cut them, cut both of them to appropriately fit one into the other. Obviously, the smaller target DNA into the bigger DNA vector that we are talking about, the thing that will carry the part that we need it to be, after all, either expressed or something, right? So yeah, we for that matter we need to enzymatically linearize the cloning vector, because generally cloning vectors are circular. So so that's the next step is now we have created this DNA construct, which is basically a amalgamation of the source DNA and our vector, right? So we call it the recombinant DNA. So now we need to introduce it into the host cell, generally bacterial cells, or in some cases phage viruses also, and or in some eukaryotic cells as well, if they are highly engineered. So and then what their purpose is, after all, to produce the protein, the thing that the for we are taking the target DNA, the reason we are taking the target DNA for. So that's the basic steps. So for that we need vehemently is restriction digestion for both cutting the vector at appropriate size, as well as the host, I mean the source target DNA from the source whole DNA structures. So as you can see, and also restriction digestion and running them to the agarose gels will give you a confirmation that if that digestion has happened, as I previously told, that we have an undigested DNA, the same thing, and then its digested version. So let's say the 2 is the digested of and undigested of, so that's how you'll get smaller versions of the undigested materials. Here also the actual size of the petty vector is 5.36, and the linear digested products is around 5 to 6, obviously. So the circular ones tend to get more on the smaller in size. That's what they tend to get. But yeah, linearized, it will be on more on the larger size. Anyways, so again, we do have only certain quantities of DNA, and we need to amplify them, right? We cannot insert a lot amount of DNA, and we cannot create and recombinate a lot amount of DNA. It can be done, of course, through plasmids, but thanks to PCR, we can amplify our gene of interest, which is generally not more than not like the whole genomes or something. It's like a part of the gene, just like the target gene that we talked about. So we can amplify it, provided we have at least one template, right? So with what we do primers, we put primers for the region that we want to amplify, and then amplify it out. So after a lot of cycles, it would be just 2 of the parent strands, and all the rest gradually it will come down to our only gene of interest that being amplified. That's why we do a lot of amplification, like 30 cycles, 35 cycles based on it. Of course, to have more of our target DNA, and at the same time to eliminate out of the initial template strands as well, in terms of probability. So and also PCR is a very automatic setup, so we don't have to. It's like the machine knows after how many seconds to move on to the next temperature. And And yeah, that's how I was telling. Cycle 1, the original strands, and from, as you can see in this region, just a second, yes, we need just this much. So after cycle, it would be like a hybrid of the parent strand and the daughter strand. And then again, after the second cycle, probably, yes, cycle, the first, only the region that we need is produced. The complete double of that region is produced. After several cycles, you'll find only two, the initial two template strands that are now paired up with newer strands will be there, and the rest all will be our whatever the size we desire, on the size of the DNA strand that we desire. So qPCR. So first of all, although qPCR, quantitative PCR is also known as real-time PCR, but just to be completely clear, it's not RT-PCR. What we generally call RT-PCR is the other one, reverse transcriptase PCR, that we use for single-stranded RNA stuffs like the COVID test. But here it's different. Okay, it's QRT-PCR or qPCR we generally call it. So here also, so the method of the template is either DNA, RNA, and real time it's used in all the formats of usages in amplifying. I mean, so here the amplification of DNA is monitored in a real time. We might need it for a lot of experimental purposes, not in our daily diagnostic purposes that we generally do, but in many optimization purposes or something. We need the real time to see how much amplification we are getting in each cycles and etc. So how does it work? So primers, in general, PCR, we don't have a primer with a probe, right? So here we do have primers with probe, with, you know, the very usual fluorescent and reporter probe kind of fluorescent molecule and the quencher kind of. So whenever the molecule binds to the DNA, polymerase expands the DNA strand, remove the probes from its reporter ion, so the fluorescence gets stops and all. So fluorescence gets started. So that's how another cycle, starting of a cycle, is determined. So finally, the probe is cleaved and the dye is released, and that's how the reporter molecule emits fluorescence, right? In general, fluorescence that we are talking about. So here the primer that is attached with the probe and the reporter ion, which after being released, the probe being cleaved, emits the fluorescence. So that what we do is that fluorescence is monitored, and based on when it is happening, we do get a curve with respect to the time to monitor in the real time. Okay, so now RT-PCR, what we were also talking in the, I guess, the previous session. PCR is reverse transcriptase PCR. So again, QRT-PCR, which nowadays, just to avoid confusion, are called qPCR, quantitative PCR, and not real-time PCR. Both are the same thing. But RT-PCR, when we call about it, we call about the reverse trans PCR, or the reverse transcription PCR. So as the name suggests, reverse transcription. Here, why do we need to do it? It's just a first initial steps that is different. That is, we do have our template DNA, the template nucleic acid material as the RNAs and not the DNAs. So for a normal PCR, we do require DNAs because that's how the whole all the setup is being made, like annealing. First, the two strands are denatured, then primers are annealed, and then elongated. Again, it is joined, and then again the whole thing gets repeated, right? So we do require double-stranded DNAs for that matter. So here we do start with mRNA or RNA. Obviously, we would carry this out with mRNAs only because that's where the information lies. So of all the RNAs that are present. So we convert it to cDNAs. What are cDNAs and what is the difference between cDNA and DNA? It's simple. cDNA is just the coded part of the DNA. So we know that in our whole DNA, the entire DNA sequences, in case of eukaryotes, the higher the complexity of the eukaryotes, the more there will be the things that we call I mean, the things that are not getting coded after all. It's just, according to the scientists of today's date, it might be just junk DNA or something that we have no clue of what it actually has a role of. Okay, so as of now, it looks like what can I say? Leftover product of evolution. Okay, so you can say like that. That's probably the one of the logical explanations that why we are carrying with us such a huge junk load of DNA. We probably just could have let it go. Maybe the DNA structure integrity might get lost. I don't know. So the cDNA is only, like, let's say numerically joining all the regions, only the coding part of the DNA. So that gets much shorter, and that's basically after transcription mRNA. If you already know transcription, in case of eukaryotes, you'll see there is a step of processing where the intron region of that mRNA gets spliced off, right? So that is pre-mRNA, and then after getting all the processing, splicing off of the unnecessary intron regions of the RNAs, it forms the actual processed mRNA. So this mRNA, when it would be made double-stranded into cDNA, that is only the coding part. So that's why we call it cDNA. So in RT-PCR, mRNAs gets converted into cDNA with reverse transcriptase, which basically does exactly opposite what transcription does. Transcription does what? It makes RNA from a DNA template, while reverse transcriptase is the completely opposite, makes DNA from RNA. Okay, so complementary DNA synthesis as a complementary DNA. Sorry if I have called cDNA as coding DNA. That's my bad. Although that's exactly the part of the DNA that does coding, so that's not wrong. But the naming is complementary DNA. So, the basic steps are mRNA and reverse transcriptase. It's a different enzyme, which, as the name suggests, does exactly opposite what the transcriptase enzymes will be doing. Not that there aren't transcriptase, but yeah, RNA polymerase will be doing. So, the 3' end of RNA, as we know, mRNA has a poly-A tail. After the processing, what all the three main processings that a pre-mRNA has to make a mRNA? First of all, the 5' capping, second of all, the poly-A tail, and third of all, the splicing of the intron regions. That what pre-mRNA to mRNA happens. Okay, so we are using, in this case of reverse transcriptase, we are using that poly-A tail, 3' poly-A tail, as an anchor point. The enzyme is using, not us, which allows us to use a short complementary strand of DT. So there is a long, sort of a long stretch of poly-A. Now what we are using is a short stretch of, that much stretch of poly-T, just to bind there and start the initiation of the replication of that copying of that strand on the other way around, on the 5' to 3', just the opposite. So that would create the second strand, and then again DNA polymerase would create, as you can see on the third line. So first, the red signifies here the mRNA having a poly-A tail. We give this primer of oligo-DT, which binds to this A region and acts as you can see, a primer, and elongation starts on this way around. So creating the second strand first, and then there is an enzyme that cleaves off this RNA material. I guess it's is it written? I guess this RNA is H or something. Okay, so that cleaves off this RNA part, and then the first strand eventually gets recoded, and the two strands of the cDNA is formed. Okay, so even in the previous class, we talked about melting temperature of nucleotide sequences. So it would be depending upon, like, it is necessary to design the primers, and it depends upon the size of DNA strands also, and how many base pairs are present in it. And to sort of calculate it, 4 times the number of GCs and 2 times the number of ATs. That is on an how we calculate TMs for a particular stretch of DNA or primers, when that's what we calculate it for. So just a very short, how many, what would be the melting temperature here? So how many GCs are there? 9. And how many ATs are there? I guess 11. Let's see it. Yeah, 9 GCs and 11 ATs. So based on the formula, 11 into 2, as AT corresponds to 2 degrees, and GC bonds corresponds to 4 degrees. I mean, GC pairs corresponds to 4 degrees. So as you can see, this 58-degree would be the melting temperature, melting point of this particular sequence. So by here melting point, just for the sake of reference, we don't intend to melt the whole DNA strands. We tend to melt the hydrogen bond that they would be using for pairing up with the another DNA strands. Okay, so just don't think that, okay, so in the annealing step we are giving 58 degrees, so it would melt the whole primer. No, not melting point is not for melting away the whole primer. It's just melting of that, so separation of the primer from whatever the strand it is supposed to. So that's, and so as slightly lower to that, we are expecting the primers to anneal to the DNA strand. So vector designing. So vector designing is very essential for any cloning experiment. So we have to choose, there are a plethora of vectors, plasmid vectors, and many vectors have their own purposes. Is whenever you'll need for any cloning experiments, you'll dig deep into it and you'll learn that there are cloning vectors. Yes.
Sir, Parth has a question. Parth Bansal has raised his hand.
Yeah. Okay. Yeah.
Uh,
Whoever has a question, please shoot.
I had the
Yes.
question in
Speak.
previous two previous slides.
To previous slides. This one.
One more, I guess. The one in which there were
Yes.
two strands in the primer. One more, one more.
One more.
Yeah, this one. So it's no,
This one.
no, the next one.
Huh.
So
Yeah.
here we have the two primers and we see a green section. So how do
Yes.
we know
Yes.
that the coding will stop at the green area only? Because it starts from the 3 prime and it goes till the end. So why will it stop at the green point? Well, the green blue
Yeah. Yeah.
junction.
Yeah. So I'll have to go back to this page. So imagine this region. This middle part is the green region, and the whole unending part is the blue region. Just imagine in the cycle 2, the second box, the middle two are the green region. Okay, imagined. Okay, now go to the go to the first box. As you can see, at first it's the whole lot of the DNA. So after the cycle 1, you would be finding like this uneven paired, not uneven, I mean one, the whole, the parent strand with it. From one side we have copied the rest, right?
Yes.
So in the
Yes.
second cycle, what we will have, two full strands, two partial strands, not exactly what we want, something extra, but it is
Hmm.
lesser than the original strand. Okay. So now, let's say from this side, this strand, this second strand, it is cut from the right hand side, but the left hand side, it is not cut. So now our primer will bind to the appropriate position of the left hand side. So after the second cycle, as you can see here, after the second cycle, it is cut. So you can see 2 rounds of appropriately of our size what we need, and one hybrid of the full length and the first cycle में जो हमने जो काट के रखे. So are you getting the point? We cannot cut,
Uh,
but we can subsequently less polymerize, less elongate. Getting my point?
so like on the first cycle, the point where it stopped by itself. Like we didn't do anything.
No, 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 no. It's not. The elongation doesn't stop. We are monitoring it. We are controlling it at the start. Trying to
Yeah.
understand. So we cannot say ki stop at this position. No, it will start from where we want to. So the first cycle we cannot completely get what we want. So we will get the start right, but not the end right. So दोनों strands में एक में from the right hand side we will start good, from the and the next strand we will do from the left hand side start good, but both of them will have other end completely elongated till the end, okay, or wherever it is. So now on the next round again, then जो right से हमने सही काटा है इसमें हम अभी that will become the opposite strand now. So there we will be giving the left primer. So अभी left से भी appropriate कट जाएगा.
Oh, okay, got it.
It is already cut in the right where it is supposed to, and now the next line left से भी cut हो जाएगा, and similarly the other strand. Getting the point?
Yeah. Yes, clear. Thank you.
Yeah. Okay. So once you do now in your notebook, by seeing one of the things, imagining how a PCR would work, so you'll understand it. I cannot show right now with I don't have a pen or pencil or something, but yeah, you'll understand yourself once you do it in your own notebook or something. It's easy. Okay. So just remember, we do control where to start from, not to where to end. So the first cycle we do on one side, the second cycle we do on the other. Okay. So moving on. Yeah. So vector designing. So here we have a vector upon digestion with EcoR1 and HEI3, whatever these two enzymes are. The plasmid is of 100 kb all total, gives 2 bands as shown in the figure. Double digestion gives 4 bands. So arrange the sites of a plasmid. So we have to imagine these things are very what can I say, interesting. So as it's not mentioned that it is a linear plasmid, we have to assume that it is the general sort of circular plasmid. So on that note, if only EcoR1 produces 2 sites, so that means it has 2 cut sites, and HEI3 also is producing 2 sites, so it has 2 cut sites. So basically we have two cut sites. So you guys try this out when you have time, and then cross check it with the answers. So what we have to do is first put anyone EcoR1's का जो दोनों sides है, first put it there, and then we have to so you can see the size, right? So EcoR1 produces 70 and 30 size of fragments. So you have to cut the whole circle into uneven 70, 30, making 2. Then the HEI3 where you have to correspond to the double digestion. So what does double digestion mean again? When we are digesting the whole vector with both the enzymes. Now you can think that if we have already done once with EcoR1, no, here we are talking about 3 separate entity of vectors. So vector 1 को हमने EcoR1 से काटे, vector 2 are cutting with HEI3, and vector 3 the same vector basically, just 3 replicates of it. The third replicate we are cutting with both the enzymes, just to see what the sizes are. So these are initially in real life we generally don't do this unless we need to optimize the size and we are not clear of. Nowadays we have a whole database of what a vector, where it will cut and all. So this is how initial days is people used to figure out naturally available vectors, what their sites are, and that's how determine the sizes. By the way, you have to do the Moodle quiz that you have today. You guys already know probably by 9. So even if I keep on speaking, just do your quiz. That's utmost important. I might miss when 9 has already begun, but just go and do your Moodle quiz. You know right how to do it. So okay.
Sir, I had just one small doubt here.
So this is another yes.
Sir, yeah, sir, there is more than one way to cut the plasmid, right here. Means I can put a cut in
Yeah,
between
okay.
the two ecos.
See, we do cut plasmids by as of now, the way we do, it's not a high throughput technology. It's just using the sites of the restriction enzymes. Her
Sir, no,
restriction
I'm not
enzyme.
talking about different ways, sir.
Yes.
I'm just saying the placement of the different enzymes on the plasmid. There is more than way to do that, right? To get that same result.
You mean where
Sir,
we want to put our gene of interest?
sir, if we put the H enzyme in between the two E enzymes, such that one is 10, other is 20, and the other
So
H
what do you want
enzyme
to
will divide the 70 part into 40 and
what do you
30.
want to put in between them?
Sir, in between the
Yeah,
two,
Of course there will be. So generally we don't do have such very small set of data at hand. But yeah, if there is, there can be. So basically, you will see after all what we have at least 3 enzymes. That would be the best way, because then there won't be any confusion. There might be confusion when we are talking about only 2 enzymes. Okay, so yeah, that can happen. So these are all mentally constructed. These are not like naturally present data we are giving you. There definitely won't be 70-30. There will be 72.5 and I don't know what the 28 or something. So it won't be like this block figure. So we are just giving this so as to explain it to you how to calculate. That's it. Okay, and there definitely I don't know of any plasmids that have only 2 restriction enzymes. So there will be more. We just don't do it for the sake of not complicating it for you guys to understand. Okay, so it's just minute left. You guys prepare in the meantime for your quiz, going for the Moodle and all. And so this is another similar eco digestion, and you have to tell which lane and what fragments to be obtained, where the length in kb of all possible DNA fragments obtained. Okay, which is basically the C part. Okay, you can go and look at questions, but as of now, go to the quiz, give the quiz, come back in time, like 9:15. I guess the quiz will be over, and
You
yes,
start the session again at
you can
9:16.
rejoin. Yes, I think let's and, what should we do? Let the session be on. Just dismiss the recording, I guess, as of now. And can we pause the recording something? Do you know how
I
to?
don't think so, but I can start the recording again at
Okay.
9:16.
Yeah, we have to re-record it. Okay. Yeah, you can stop. Students, go and give your exams first, quizzes. Several generations of DNA sequencing. So the first generation was very basic for that matter. Now we know of now that the initial days of sequencing, initial days of cloning was mainly based on the plasmids and different biological constructs that we already have present in the nature, and we do just modify it a little bit, right? In the initial days, there were the least usage of machinery, high throughput technologies. So what they used to do is, sorry, extremely sorry. So they used to call it is copy or take out their the gene of interest that they want to sequence. The whole sequencing it was sort of out of the question. So even for the Human Genome Project, when it was initially started, it didn't have throughput technology that we have at today's date. So what they did is they fragmented the whole DNA sequence, the whole thing of human. Yeah, it's huge. So into several plasmids, let's say, just for the sake of easy understanding. So whatever you have to sequence, you have to first, if it is a huge, you have to fragment it down to the feasible size, let's say 500 to 1000 at max base pairs. So then you have to clone it inside plasmids, and then you have to undergo several enzymatic reactions, at least for the first generation of sequencing, just like Sanger sequence. So we have to clone it. We have to then perform the sequencing based on either termination or elongation, which is two sort of process. Sanger's are termination, Maxim Gilbert is elongation. So termination is the point where it gets terminated, and the elongation is till the point it gets elongated. This is sort of a similar on a basic similar principle, but exactly opposite principle for that, if you want to understand. So we would just looking at Sanger's a little bit in detail after a point. So this was the first generation where we needed amplification, cloning, for that matter, very, very much so, where the wet lab part of the experiment was very highly required, and any mistake over there would refer to a huge mistake to the point that we won't even recognize. Next came the second generation, now which we call as next generation sequencing. It's a high throughput technology where we don't need plasmid kind of stuffs, where we can run so if you are doing plasmid, you cannot do much more replicates of the same thing, right? It would be really implausible that you'll do so many of plasmids and then do of all those sequencing. So if it is just a one-off incident that your sequence is this, or if you are having significant sample number, sample size n, a significant n, so that you can confirm that okay, the sequence that I've had is correct, right? So after all, because that came out from wet lab experiments, so that had a lot of chances of getting what can I say, errors. But here that error chance sort of gets reduced, and we can take a more number of sample size n for that matter. And here majorly the machine does the work. So the genomic DNA fragmentation and then ligation, it is done with probes. It's not based on the termination or something. And also, yes, in the first generation sequencing, after we got the products, we had to run it on gel so as to know where is each strand. I mean, after the terminations, where do each strands lie in terms of the size. So that was also a constraint. Here it is all done high throughput in the softwares. So the softwares analyze it, and also the visualization there was through ekdam wet lab physical gel. Here the visualization, here the reading is done through lights. Okay, so the size of it is determined, size and what it is ATGC may say which one, it is determined by lights and the differential fragmentation of light fragmentation of each of the ATGC, and that's how the sensor, the biosensor that detects it, the detection sensor detects it, either if it is a ATGC and if it is 2A, 2G, all sorts of things. So there are various like in today's date, Illumina, Iron Torrent, and I guess Solid also. These also are basically second generation. They are a little bit varied in terms of what they do and how they do. You can go on and read about it, but it's really high throughput. So and the stitching is also done by the software. Stitching, what stitching? So even this that cannot do like the whole DNA. It would take a little bigger fragment in each case, but nothing can do like start from one end of the DNA and entire complete. So yeah, that we will come. So that cannot be done in this too. So this also does with fragments, and this stitching is done by the software. So we don't have to take the pain of stitching in the sense. So let's say fragment one we get this is ATGC, GCGC, whatever sequence, and then fragment two we get this. So there are overlaps. So those we manually had to when it was first generation, or we had to create our own sort of program so that we can find overlaps and then join those fragments and make the actual bigger DNA or nucleic acid for that matter. Here, all these things are done by the software. So obviously humans made the softwares. So, but anyways, it's the each and every individual human doesn't have to take the pain of it. Third generation sequence is basically where you can sequence an entire single like a whole DNA molecule, and also it can sequence like single nucleotide it can detect. So it's totally a new principle of the nanopore technology that we call it, that is completely new, although it's not it can do a lot longer sequence, as I told, entire DNA molecule, one single molecule, like you can identify one single molecule and go by one single nucleotide basis, and it is getting even more popular these days. But the number of replication, again, it's like one after the another. What the thing of NGS is, you can do a lot of replications means the sample size n, you can do that lot in a less time. But here you take time, but the pain is even lesser, and the chances of error is even lesser in case of third generation. Okay, so moving back to the first generation, how it all started. Sanger sequencing being one of the most popular and one of the most well-known. Okay, both are the same words I used. So it has basic 7 steps. First of all, so these 7 steps we are talking about only the sequencing part. The prior to all those cloning warning is done. So now we have each fragment. So now starting with the fragment, denaturation of So denaturation, again separation of the two strands. Then we have single-stranded DNA. Okay, so now we are amplifying the single-stranded DNA, attaching a primer, and now we have four polymerase solutions. So what they will do is it will grow until that polymerase is it. So it's like ddNTP, as you can call it. ddNTP is, it will attach it would supply that dNTP. Okay, so let's explain. Let's say the purple one here in the test tubes, the purple one is the DATP. So the we need dNTPs, right? Like ATGC nucleotides. So that will go and be the bricks of this growing DNA strand that we are polymerizing, right? That would be those individual ATGCs that will bind over there. So now in general ATGC, what is the thing? They have a loose hand so that they can bind to the next ATGC, whatever the nucleotide is coming on the next in line. But when we give a DDNTP, like dideoxy nucleotides, they don't have that free hand. So basically, no further nucleotide can bind after them. So they are the stopping points. Okay. So let's assume the violet is the one having DATP, like the modified version of A. So and the other things are basically same, the normal G, T, and C. Okay, but the A over there is modified. So whenever there is an A in the growing chain, whenever there will be an A, it will stop, no further going. Now you might say there might be multiple A's in a particular sequence, and so the fragmentation, as I already mentioned, so it also happens in a every every nucleotide, it gets fragmented. So we have several fragments. Let's say it's a 10 nucleotide chain. We have like 10, 9, 8, 7, 5, 4, 3, 2, 1. So we have so many fragments of it. So each fragment will start and end wherever there is a A. So let's say the one has one or let's say 10 nucleotides. It will start from here. The first one will get in. The one which is already 6 nucleotides grown, it will from, let's say, there and end to the next one. So sort of like that. So the chain grows until termination with the respective fragments, and the resultant DSD double-stranded DNA fragments are denatured to obtain a series of single-stranded DNA variations, and finally they're separated through electrophoresis, and the sequences interpret. So here is another one example of it. So you sequence a single using Sanger method. Below is the resultant sequencing jail. What is the sequence of the original strand? Now, moving back to this, you can see the 3 in the 3 number image. The red strand is the original strand, okay? And the blue strand, that is where you are putting the primer, all the growing is going on, okay? So that is the one that is being coded, and that is the one that you'll be putting into the gel. So whatever you finding the sequence present over here is of the blue. So you have to do the complementary to find the original one. Blue strand was coded, okay, the red ones. So here, as you can see, so DDA gave the lower gel bands are, what does it signify? It signifies the least size, right? Smallest size, right? The higher the strand, it is size. So the smallest size means what? The start of it, the start of the sequence, right? 5' end of the sequence. So from 5' start this A, G, T, C, A, then again 2 G's, then C, then A, then T. Be from 5' to 3'. Yes, but this is which? The blue strand, which the complement of which will be the actual original red strand, which is this one, exactly opposite. So see, TAC and start with ATG, and then TGA on this front, it would be ACT on the opposite the of this. So this is basically the original red strand from which the blue strand is formed, and then and it is differentially formed. So that's how we can understand start nucleotide, which one is stopped. So D has stopped on this final D has stopped on this, I mean, whatever the lesser fragments we have created, and D has stopped A as well. Sorry, I mean A, sorry, my bad, not D. DDA has stopped on these three. So first of what we can assume that, okay, there are three G's, there are three A's, and there are two T's, and there are two C's. Where the position, that would depend upon the size that it is accumulated, size it is showing. So the least size is A, then the two nucleotide size is G, three nucleotide size is ends with T, four nucleotide end with C, five nucleotide ends with A, so on and so forth. So that's how you can line this up, and the complementary will be the original. Okay. So this is again we have what we call it, a template strand given, and would be the one. So ideally it will be the 5-1 having five T's present over here, B, but be careful of what it is asking for. And I mean the one that we are actually coding, or the original strand from which we are coding. So based on that, the things will be different. Okay. So if this was this we were calling about the original strand, then there would be the opposite. The A's will signify how many T's are there. Okay. So here the DTTPs are used as dideoxynucleotides. So yes. Okay, so all these sequencings, why? Just for the sake of fun that we do? No, definitely not. We are not doing this to know, okay, how many A and T and G and C's we have in our sequences, or what other sequences. Why do we need to do sequencing is of many of the interesting things that we can do. One is to understand, obviously, evolution, lineages. So be that imminent lineage, like our parental heritage sort of lineage. Yeah, you can do those parent animations through today's days sequencing. Yes, we can. And also, if we can do this, we can also do where our ancestry, like, so, and also the proof of human migration. I mean, where it all spread, how it all spread, from which subspecies, I mean, from which different kind of our ancestors mixed with which different kind of, so on and so forth, and a lot of things. Just not for humans also, it applies to anything having a TGC, right? So just here as an example, Neanderthals, there was a study carried on, and it has been found that they have contributed to 1/4 of the DNA that we Eurasians have. So this has been concluded. The whole, obviously, the whole genome sequencing and specific sequences matches with. So how it is done? Like what is evolution, or this changing of different species, basically changing of the nucleotides, as even in COVID you might be hearing it is mutating. So what is basically, it's DNA sequence is getting changed, either change, it's a formation of the final protein that is getting changed. But as it's a virus, it's obviously the DNA getting changed because it doesn't have a further get changed, right? So everything that is has is that single stranded RNA in case of this COVID virus. But in case of ours, why don't we change as much as that? Obviously we are not, first of all, single stranded, that's a huge point, but also we do have several levels which was prepared not to change, which were prepared to, while that of the viruses, because they are single stranded, their strength is number, our strength is individuals, so they have a lot of number. Let's say we are targeting their virus, I'm talking about right now, we are targeting their spike protein, so the ones that have a modified spike protein, and the ones that do not, they won't. That's it. The number, they are playing the number game. We are not. Obviously, we won't afford to lose a lot of compatriots just because they don't have that DNA, because we are social animals, and also we cannot, because we have several stages like DNA, which is very I mean comparatively, then RNA, and then so on and so forth. But in due course of time, like millions of years and thousands of years we are talking about, yes, there do changes DNA, and the amount of change corresponds to how far we are from the one on the another lineage. So jitna less change, that means they are nearer to us in terms of the hereditary or lineage that we are talking about. Okay, so yes, this was of that. And there is a, sorry, cloning, you have a endangered endangered panda species, and you want to preserve it. So what you will do, you take the oocytes of the genetically unimportant animal, let's say of a similar kind of panda, but you want just a particular lineage of the panda, you want to reculture it and keep it alive basically. So you take the oocyte of some similar animal, let's say here we have a different species of panda, and then we genetically inject the nucleus of the one we want by removing the nucleus of the one we don't want. We are just taking the oocyte of that particular panda, of the not so important animal. And then now we have basically the one that we want transferred into the womb that is present, and we can have the species that we want. So that's how preserving embryos, preserving the genetic even the cell lines, basically preserving the chromosomal set, the nucleus is very important, and that we can do by preserving the cell lines and the germ cell lines even also. More importantly, yes. So yes, we have surpassed quite a lot of minutes. Sorry for that, and I hope you had good time learning a lot of new things. Any questions, please? If you have, you can raise hands. Okay. I don't see much hands. Dipri, are you done with the necessities that are needed to be done?
Yes, I recorded the attendance.
Okay. Okay. So I guess
Yeah, I think we also have a class from 9:30, so we can end this session now.